Hi everybody, and welcome to reviewing the metabolic processes, cellular respiration, and photosynthesis. We know that for cellular respiration, glucose and oxygen are required to produce carbon dioxide, water, and energy. Let's start in the cytosol of the cell with the first step of cellular respiration, glycolysis. 1 ATP is used to convert glucose into glucose 6-phosphate. An easy way to remember this is the fact that because ATP loses one phosphate molecule and becomes ADP, and that phosphate attaches to the 6th molecule of carbon in glucose. Next, glucose 6-phosphate isomerizes, which is a fancy way of saying rearranges into fructose 6-phosphate. Then another ATP is used, which means fructose 6-phosphate gains a phosphate group and becomes fructose 1,6-bisphosphate. This breaks down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which are isomers of each other. And since cells prefer G3P over DHAP, it is converted to G3P. Hydrogen is released from G3P and a phosphate group is added. This produces 1 NADH and G3P becomes 1,3-bisphosphate glycerate. Now that the molecule has two phosphates, one is released to produce ATP. This converts the molecule to 3-phosphoglycerate, and again, since cells are picky, this rearranges to 2-phosphoglycerate. Then through dehydration synthesis, which means water is produced, this turns into phosphoenolpyruvate. Then in the final step of glycolysis, the phosphate is released to produce another ATP, and the final product is pyruvate. Even though the total amount produced is 4 ATP, the 2 ATP used in the beginning stages make the net amount of ATP as 2. The next step of cellular respiration is pyruvate oxidation. This occurs in the mitochondrial matrix, the center of the mitochondria. This is a short process where each pyruvate is decarboxylated and an H ion is released to produce NADH. Then, coenzyme A, also known as CoA, is added so that the end product is acetyl-CoA. The process occurs twice because 2 pyruvates are produced for each molecule of glucose. The next process, which is also in the mitochondrial matrix, is the Krebs cycle. This starts when acetyl-CoA joins oxaloacetate to produce citrate. This isomerizes into isocitrate. Next, an H ion is released to produce NADH, and isocitrase is decarboxylated into alpha-ketoglutarate. This molecule again is oxidized to produce NADH and decarboxylated and reduced by CoA to produce succinyl-CoA. Then the CoA is released to produce succinate. This process creates 1 ATP through substrate-level phosphorylation, where GDP becomes GTP, which is converted to ATP. Succinate then becomes fumarate, which releases an H ion to produce FADH2 and becomes malate. Malate releases an H ion to produce a molecule of NADH and becomes oxaloacetate. And the cycle repeats one more time for the second acetyl-CoA produced by one molecule of glucose. The final stage of ATP production is the Electron Transport Chain. This is in the inner membrane of the mitochondria, or more specifically, the cristae. The electron transport chain consists of 4 proteins labeled as complex 1, 2, 3, and 4. The complexes are arranged from lowest to highest electron affinity. The chain starts at complex 1, where NADH releases the hydrogen and the electrons enter complex 1 to go through a series of redox reactions. The energy produced from the redox reaction causes complex 1 to pump out the hydrogens from the mitochondrial matrix to the intermembrane space. A similar process occurs in complex 2. However, it occurs with the electrons from FADH2. However, complex 2 cannot pump out hydrogens to the intermembrane space, so the hydrogen ions remain in the mitochondrial matrix. Once the electrons have gone through complex 1 and 2, coenzyme Q helps transport them to complex 3. Once again, the electrons move through redox reactions, which provides the energy required to pump hydrogen ions into the intermembrane space. Cytochrome C helps transport electrons to complex 4, and the same process occurs once again. The electron transport chain concludes with oxygen, the final electron acceptor. Now you may have noticed the intermembrane is quite saturated with hydrogen compared to the mitochondrial matrix. The concentration gradient causes facilitated diffusion through the ATP synthase. The energy from the hydrogen going where it wants to go turns ADP into ATP. The facilitated diffusion has a coupled reaction with the final electron acceptor to produce water. So when we look at all the processes of cellular respiration, we can see the ATP produced by each stage of cellular respiration and how the energy carriers are used in the electron transport chain. There are 36 or 38 ATP produced per molecule of glucose. The most ATP is produced in the electron transport chain due to the oxidation of the energy carriers NADH and FADH2. NADH produces 3 ATP in the ETC because it releases 3 hydrogen ions. FADH2 produces 2 ATP in the ETC because it releases 2 hydrogen ions. We can also see the other products of cellular respiration, such as water and carbon dioxide. Through the products of the steps of cellular respiration, we can see how the formula of cellular respiration applies. Let's talk about photosynthesis in plants. In this case, the chloroplast is the chef of the cell, and the organelle that provides the food for the plant to survive. The important parts of the chloroplast to know are the stroma, thylakoid, and lumen. Photosynthesis starts with the light-dependent reactions, which occurs in the thylakoid membrane and the lumen and stroma. This starts when photons hit photosystem 2, exciting the 2 electrons in chlorophyll P680, transferred to the plastoquinone or the PQ, an electron carrier. Then they are transferred to the cytochrome complex, which uses the energy from the electron to pump 4 hydrogen ions from the stroma to the lumen. The electrons reach photosystem 1, where they are hit by photons once more to become excited again. They pass through the electron carrier FD and reach the enzyme NADP reductase, where the final electron acceptor NADP is reduced by 2 electrons and 1 hydrogen ion from the stroma. Because the electrons in photosystem 2 were transferred to the electron transport chain, protein Z breaks down water into 2 electrons, 2 hydrogen ions, and oxygen. The electrons from water replace the electrons transferred in photosystem 2, and the hydrogen ion remains in the lumen, and oxygen is a byproduct released through the stomata. This results in a concentration gradient of H ions between the lumen and the stroma, which provides the energy required for phosphorylation by facilitated diffusion through the ATP synthase. The chemiosmosis produces 1 ATP for every 4 hydrogen ions diffused into the stroma. The next part of photosynthesis is the Calvin cycle. Unlike the previous stage, this cycle is light independent and occurs in the stroma. It consists of 3 steps: carbon fixation, reduction, and regeneration of RuBP. It starts when 3 carbon dioxide molecules enter the cycle and are attached to 3 ribulose bisphosphates with the help of the enzyme Rubisco to form 6 3-carbon compounds named 3-phosphoglycerate or PGA. Then 6 ATP are used to add a phosphate group to the molecule, producing 6,1,3-bisphosphoglycerate or 1,3-BPG. This is reduced by NADPH and a phosphate group is released, producing 6-glyceraldehyde 3-phosphates, and then 1 G3P exits the cycle to be used to produce glucose. The remaining 5 G3P are converted to 3 RuBP using 3 ATP. Then the cycle keeps repeating to produce more G3P. If we think back to cellular respiration, we know that 1 glucose is made of 2 G3P. This means that this cycle needs to run twice to produce 1 molecule of glucose. We can see how the reactants and products of photosynthesis results in this expression. When comparing cellular respiration and photosynthesis, we can see they have quite a few similarities, such as they both include an electron transport chain, they both have a cycle such as the Krebs cycle and the Calvin cycle, both processes use phosphorylation to produce ATP, and also include the use of hydrogen ion gradients. They also differ in the fact that photosynthesis requires light, and these processes have opposite products and reactions. Thanks for watching, and I hope this video refreshed your knowledge.